Ribosomes, site of protein synthesis. So this is where the messenger RNA will go in the cytoplasm, whether it be on the rough endoplasmic reticulum or on free ribosomes. So mRNA, and we get those down. mRNA is messenger RNA. Makes the copy, remember, AUCG now, AU, not T anymore, CG, off of the DNA. And it takes it to the ribosome. This is where the machinery happens, where you start building proteins. So the ribosome has two subunits, a large and a small. So we know about this, where they are. The RER are free in the cytoplasm. And ribozymes are enzymes that are needed to build these proteins, the developing proteins. So you have a small unit and a large unit. It's like a ticker tape machine, if you remember what that is, where the messenger RNA goes in and it kind of moves through, and you're building proteins based on building amino acids, I should say. First, you have to build the amino acids with the nitrogenous base codons from messenger RNA, and they pair pretty much the same. The endoplasmic reticulum is membranous, like the nuclear membrane is continuous basically with the membrane of the ER. So the rough or granular ER has the ribosomes that are studded or embedded on the outer surface. And it builds proteins or modifies proteins as well. The smooth ER does not have ribosomes. It's agranular without those granulations of ribosomes. And each cell will be different for what it needs to make, like carbohydrates, phospholipids, steroids, anything that's not a protein. So basically we saw this in that nice Hyundai cell before. Hyundais are actually pretty nice, some of them, with a lot of options. Gotta find a new car. Okay, Golgi complex. Now this is the post office of the cell, if you will, from 7th grade. So I like to call it the Golgi apparatus. Again, it's a membranous structure. And the membranes have little caverns called cristernae. So one side receives input from the ER, and it packages these into vesicles. Vesicles are always like a part of the, well, they're derived from the plasma membrane, so they have their own membrane, vesicles, like little spaceships. So the plasma membrane buds off to form these vesicles, and then if it's something like a neurotransmitter or something that needs to leave the cell, the vesicles will spit out that content through the process of exocytosis, the opposite of endocytosis, which is more like phagocytosis. So proteins are modified within the Golgi apparatus, and other things, not just proteins. Retrograde means opposite direction. So anterograde is one direction, moving forward. Retrograde is usually backwards or back into. So extracellular proteins are brought back into the GC, or the Golgi complex, and they'll package the amino acids and have them be sent back into vesicles, whether sent out of the cell or used somewhere else in the cell. So I guess we could put like a picture of what they are, so you know. I don't know if you're doing this type of thing in lab, but at least so you understand what all these cells are going to look like. So when we get to the specialized cell, you'll be ready for it. Okay, so I think that's most of them. So the nucleus is, of course, has much more going on. So this is more like the government of the cell. So let's go through that. So muscle cells, especially, really I should say skeletal muscle cells. Skeletal muscle cells are the voluntary muscle type that move our joints and bones. They have many nuclei, multinucleated. And cardiac cells may have one or two per cell. So mature red blood cells, called erythrocytes, are what's called anuclear. You see an A in front of something, it's usually not there. So the nucleus has a membrane, which we call the nuclear envelope, and it has the nuclear pores. So the outer membrane, like we said, is continuous. The membranes are continuous with the rough ER. And the Golgi apparatus has a membrane, and the smooth endoplasmic reticulum has a membrane. So the nuclear pore complexes, which allow small molecules like RNA, and of course ribosomes, move in and out of the nuclear pores. This should be a close-up here of the nucleolus, which builds ribosomes and builds ribosomal RNA. And these are your nuclear pore complex, just big enough to fit these small molecules like RNA and ribosomes. So DNA, we talked about this at the beginning, and genes. So this is where DNA lives, all right? A gene is a strand of DNA, long chain of DNA. And along those genes, or in each gene, it codes for a specific protein. Thousands of these we'll see that code for proteins. So the pairs, A, T, C, G, building amino acids that are building protein based on your genes. So transcription, this is important. Transcription is the messenger RNA making a copy of DNA in the nucleus. So transcription happens in the nucleus. Please remember that. And then mRNA could leave the cell with that information. ATCG or AUCG now that it's RNA. So it's sent to the ribosome and it's translated into the appropriate amino acid sequence to build proteins. And transcription and translation, now translation happens in the cytoplasm. Where? On the ribosome. So by all intents and purposes, translation from messenger RNA to amino acids is protein synthesis. So transcription is in the nucleus. That would be a good takeaway from this part of the lecture. Nucleoli, that's the nucleoli, and that contains DNA that codes for the production of ribosomal RNA. So nucleolus is building ribosomal RNA, which is building ribosomes. That's rRNA. So we have about 25,000 different genes. And the genome is all our genes. So again, the DNA will make up genes. Genes are on chromosomes, and sometimes they look, a lot of them look like X's. You know, so the chromosome contains different genes, right? And there's 25,000 that we recognize on different genes, so on different chromosomes. So I'm just gonna tell you this: a somatic cell, anything besides a sperm or an egg, has 23 pairs of chromosomes. Okay, and we have what's called 22 autosomes, and I should say 22 pairs of autosomes, and one pair of sex chromosomes. And that basically determines male or female. So the one pair could either be two X's for female, or X and Y for a male. So total chromosomes, how many chromosomes do we have in the nucleus of one cell? There's 46, right? So there's 46 total chromosomes. Sorry, chromosomes. And we call that cell a diploid because it has those 23 pairs. So there's 22 different autosomal chromosomes that have different genes on them. They're what's called homologous, which we'll see later. They basically have two sites where the genes are and one pair of sex chromosomes. So these also, the sex chromosomes also carry genes. That's why, like, if something like, and you guys getting this? You guys kind of understanding this? Like the X and Y and XXY. So like, well, just to get to know what genetics, how this works. And this, believe me, this is not a genetics class. It would take 3 lectures just to even introduce the topic. But the female has that one pair of chromosomes is 2 X's. And the one pair in the male is X and a small little dinky little shriveled up little Y, right? Which basically makes them male, androgen. So like if you have a mutation or a genetic problem, like say color blindness, you've heard of color blindness, right? Where you can't really distinguish between green or red, I believe it is. I don't have that. So this is a sex-linked condition, meaning that it's linked to the genes on those sex chromosomes. So if the female has, and it's dominant, so which means that, you know, you have to have the dominant trait on at least one of the chromosomes to have the condition, the sex chromosomes. So let's say that the lady has the dominant gene on one chromosome, but her other chromosome does not, those 2 sex links. So the one chromosome kind of hides the gene. It doesn't express the gene. But the male only has 1 X chromosome. So if that dominant gene is there, he's going to get that condition. So some sex-linked conditions are more prevalent in males, like color blindness. And it's because of that, they only have the 1 X, so you can't really suppress that gene. It's going to express itself in its phenotype, which is the expression of a genome. So girls, that's pretty good, right? Less chance of getting that. And and muscular dystrophy too, which is another horrible condition. Getting phone calls like crazy. My phone never rings. Maybe it's Popeyes. The delivery's here. Hold on, I gotta go get my food. So any questions on that? They're canceling schools tomorrow. Not yours, though. Let me know if you get in, even though we're all online. Your school for my classes, anyway. Poor Professor Shivani. She'll have to, she only has Monday labs. Okay. So again, this is a little complicated, so I want to simplify this as best I could. So again, diploid cells, 23 pairs of chromosomes, 22 autosome pairs, and 1 sex chromosome, the termination of sex through that Y chromosome. So messenger RNA is altered after it's transcribed. And where does that happen? That happens in the nucleus of the cell. So DNA replicates. Messenger RNA makes a copy of that. And then it leaves the cell, right? So it's building proteins. Proteins are many polypeptides. And the polypeptide bond is between amino acids, like amino acid. That's a dipeptide, and this is a covalent bond. But in proteins between amino acids, we call that a peptide bond. So amino acids are built based on the ATCG or ATC or AUCG. And it's based on 3 of those bases to build a specific amino acid. So proteins can be modified very easily. And this kind of, think of this a little simpler. And Chris was talking about this before, which is kind of cool. Like how the expression of a gene could be either turned on or turned off based on maybe your lifestyle. So here's a question for you at this point, just to get, before we get into this heavier topic. Raise your hand virtually, because I can't see everybody, if you believe that what's in our genes makes us who we are, basically 100%. Okay. I see a couple. I see a couple. Who thinks that what we are almost 100% is based on our environment or where the stork drops us? You know what the stork is? You know what that is? Yeah. Where the stork drops you. That's how we were born. Like where we live, our lifestyle, our environment. Who thinks that? That's mostly that. Cool. Hands are up. Virtual hand. Who thinks that it's kind of a nice combination between the two? Genetic. Oh, you can only raise your hand once. Genetic. Yeah. So that's A. Chris gets an A. Tina gets an A. Let me just put this in the grade book. Lexi, Cal, they all get A's. Yeah, that's true. Don't you all agree? So if you're carrying a gene, the expression of it is not necessarily your fate. So if you have a gene that we discovered that could lead to diabetes mellitus type 2 or Alzheimer's or ALS, unless it's a hardwired gene, for hair color and all that kind of thing. For these proteins are very mobile, and the expression of them could be turned on and turned off. So it's not your fate because you have a specific gene. So does that make sense to everybody? So you'll hear words of modifications of genes like methylation, phosphorylation, and splicing, right? Splicing is cutting into small units that change the expression. Sometimes it suppresses a gene. Like now, like I'm saying, like we could be sitting here and there could be a gene. I don't mean to be depressing, but there could be a gene that could lead to an illness. I'll just say illness because that sounds better. Sounds more textbook. And that gene could be suppressed, not only by your immune system. I'm not even talking about immune system. I'm talking about the way this machinery going from the DNA of transcription into translation. Because proteins are very mobile. They're being built all the time. New proteins being suppressed, being destroyed, being built, being used. Enzymes being made, destroyed, and expressed. So that's the concept I want you to leave here with. You don't have to know everything about the enzymes that split DNA or cause the building of polypeptides in the ribosome. They do, of course, the enzymes are extremely important. And without those enzymes, it's not going to happen, or it's going to be delayed or mutated if there's a problem with the enzyme. Remember, enzymes are proteins. They have to be made and transcribed in your nucleus and then translated on a ribosome. So proteins are very important on how they're made, and if they are transcribed into protein, is that gene going to express that outcome? So keep it as simple as you can. I know that's confusing. So chromatin, like we said before, is the DNA within the nucleus with proteins called histones. And they're going to organize these chromosomes when it's time to undergo mitosis. So histones get positively charged, react with the negatively charged DNA, causes that coiling, right? Coiling, and they're held together by hydrogen bonds. That's an interesting point here, right? When we're talking about that polarity, right? And the particles within these DNA are called, within the nucleus, called nucleosomes. So that's basically just a chromatin for our purposes here. And we'll get to what chromosome is. And a chromosome, as you know now, is a group of genes, and the genes are made up of strands of DNA. So the chromosomes don't leave the nucleus, right? Just the messenger RNA leaves the nucleus. So euchromatin, you usually mean like a EU, usually responds to self or familiar or like or normal. Sometimes it's normal. It's perceived as normal. So the chromatin, which is DNA, is active in transcription. And it's looser, and chemical changes and acetylation, which allows it to be expressed. normally. Like, again, hardwired genes like, you know, your earlobes attached, or you can have a, what do they call that, a widow's peak, or brown eyes. All those things are pretty hardwired as far as genetics go. But enzymes and protein receptors in cells are not. They're always changing in the expression to the way they are made. So chromatin is good for those kind of things, but you got to be careful in science when they try to kind of manipulate the genes to all look the same, right? That's why cloning is so controversial sometimes, is you're going to build the same, you're going to lack diversity. If you lack diversity, that's when the species is at risk. So chromatin is important inside the cell, but not in science. I mean, it's important to learn about it, but not to use it, of course. Okay. Anybody have any questions about that? That might have been confusing. Anybody kind of understand what I'm talking about there? So the basic takeaway from that is that you need diversity in these genes, and that's what makes them stronger. All right. In reproduction as well, for any species. So heterochromatin now is inactive regions of the DNA, so less expression. So it seems like right now, like even in science, like a lot of the genes or the DNA that we have is not really used. But eventually we'll figure it out. We just see a lot of the DNA that we have in our cells is being kind of wasted and reproduced. So that's another fact that it's good to know. Kind of like the oocytes in the female ovary. Like when you're born, night before you're born as a female, you're making millions of primordial or follicles or immature oocytes, but you really only get 400 or 500 usable oocytes in your lifetime. So that's about reproduction. So DNA probably has the same evolutionary balance. So we're just on the surface of it, really, but we're getting there. So DNA, and here's your base sugar, phosphate, nucleotides, and the hydrogen bonds will coil around, forming this double helix, which is DNA, and the histone proteins will kind of be wrapped around that, and eventually they'll thicken. And this really only happens like in one part of the cell cycle, let's say in a day. And then they look like this. This is not the X chromosome sex cell. This is just any chromosome, any autosome. And we have 22 pairs of these and one pair of sex chromosomes that kind of look like that. So the genes are all along these loci or locus, locale, if you will, along these chromosomes. And there's 2 of each. So one gene could be expressed or suppressed based on which chromosome it's on. So again, this is just showing you really don't have to know acetylation. But I want to talk about transcription factors. All right, transcription factors are part of the proteins that are built on the ribosome and also transcribed from the DNA through messenger RNA. So they call this epigenetics. Let's put this word out there, epigenetics, not eugenetics. That's not good. Epigenetics. So we're learning about this in the last decade or so, about how a transcription factor could either cause the expression of that protein being built or the suppression of that protein being built. So again, this is gross examples, but you know, say eating superfoods can cause a suppression of a bad gene and an expression of a good gene. So lifestyle could matter here. So this transcription factors being turned on or turned off, maybe turned off because of too much inflammation or certain foods you eat, or stress, or lack of sleep, or exposure to environmental toxins can affect the expression or suppression of a gene, whether it's going to help you or hurt you. So that's really important. That's a study of epigenetics, is the study of these transcription factors that either express or suppress the gene that's going to express a protein. Yeah, right. So this is basically back to the nuts and bolts of how the machinery works, where it's DNA directed to the RNA. So RNA is synthesized and copied from the DNA, and that's called transcription. So this is a sequence of what's called codons, which are 3 bases, basically, forming together. So the 3 bases, like a GC, for instance, will form an amino acid based on that. So you don't have to know about promoters and the parts of the DNA that start or stop these codons. But it's all based on these base pairings from the nucleotides. So transcription factors bind to the DNA to begin transcription or stop it. It depends on if it's suppressing or enhancing the expression of that gene, expressing or suppressing. I like that too. So whenever you see ACE at the end of a word, that's an enzyme that helps break bonds. In this case, it's breaking the base pairs of DNA, because DNA has to split, so RNA could be copied from that DNA. So remember, DNA is CGTA, and then you add the U on the RNA, and that leaves the cell. So remember this, RNA has uracil instead of thymine. Okay. So only one freed, that is a word, DNA strand is transcribed. So the DNA is going to replicate, and one of those, and then it'll split. And when the 2 strands of DNA split, that's when RNA is transcribed from that piece that was synthesized. And then there's always the DNA, let's say the 1 strand stays in the nucleus. And all DNA stays in the nucleus. So, okay, so that's as deep as I want to go on that, because we could really go crazy with this. So, types of RNA. Messenger RNA, precursor to messenger RNA. And that messenger RNA is made by transcription. Then it can be modified, right? And that's the modified version of messenger RNA is what goes to the ribosome to build the protein based on that template that it was transcribed in the nucleus from DNA. Transfer RNA is in the cytoplasm, and that will organize the amino acids, the free amino acids, like the inclusions that are in the cytoplasm, and bring them to the ribosome. So messenger RNA can translate those. So basically the transfer RNA is bringing in the materials, and messenger RNA is the architect to put those together, those base pairings, to make amino acids. And then the ribosome continues to spit that out with peptide bonds into building either polypeptides or proteins. A lot of things about protein, a lot of words about proteins, right? Ribosomal RNA is really the ribosome. So the little bit of protein plus the ribosome forms the ribosomal RNA form the ribosome. Okay, and that acts as an enzyme, and enzymes are protein, right? So again, I don't want to get too far into this with introns and exons, but the messenger RNA, as it's copied from the DNA, there's a lot of extra DNA, like when it's kind of snipped out with enzymes. So the introns are not expressed. They're not the part of the DNA that's going to be usable. So exons are the ones that are spliced together, and they're going to ultimately make up the genes. So introns are cut out. Exons are spliced together DNA chains. Really, it's RNA at this point to make those amino acids. And then they're spliced. And this is a cool word. I just like to say SNRPs. So the 2 parts of the RNA, it's really DNA replicated with uracil, are spliced together with these SNRPs, small nuclear ribonucleoproteins. SNRP, so that's a cool word. So that's the splicing of the messenger RNA. This might show it a little bit here, how this is spliced. And the introns are pretty much spliced off, and the exons are spliced together. So what happens to the introns, you might be asking? Well, it's just recycled, broken up, and used as nucleotides, as far as we know. Is there anything else? we're still finding out. So it seems like there's a lot of extra DNA that's not actually expressed on these human chromosomes through the genes. Not the genes like Levi's genes, of course, the genes on the chromosomes. Okay, now this is really cool. Two ladies, I always forget their names, but they just won a Nobel Prize in science, genetics, for using what's called CRISPR, clustered regularly interspersed short palindromic repeats. So just remember CRISPR, right? And they found it with using bacterial DNA. So again, it might sound unethical, but their plan was to be able to locate a dangerous gene that could express something like Alzheimer's or sickle cell anemia or color blindness or ALS, and splice that gene and remove that gene, either from the parents, the fetus, or the person that is prone to have that disease genetically. So they use this CRISPR gene that actually splices it, and they got it from a bacteria. We learned it. So it actually can be something used in medicine. So now, why this is important. Everybody okay? I know this is getting a little intense here. You guys okay? So Shana, you good? So now when you go to the doctor, or hopefully in the future when you go to the doctor, they just won't look at the disease or look at the symptoms and then, okay, this worked for 100 years. Let's do this. But now, before you're even born, they may be able to tell the predilection for something and the statistics of what the chances you may have of having, say, diabetes mellitus type 1, either at a very, very young age or before you're born, or based on your parents' genetics. So it's kind of making sense. So that there'll be more individualized medicine based on these genes that are showing. Maybe even the bacteria in your body. Maybe even what's on your chromosome as far as expression of a gene or phenotype or genome. Any questions on that? That's a little, I just want to get that concept across. So this CRISPR is that DNA being used to actually splice our DNA to pretty much remove a gene that's going to cause something that could shorten your life or cause a disease. I think that's the best way to say it. Okay, so it could inactivate the gene. Okay. You know, and so they've been working on this for years in other mammals, of course. So now they tried it in humans, and it actually can help people with the diseases or predilection to the diseases. Okay, how are we doing so far? Okay, so now protein synthesis is translation. So I'll just get into this a little bit, and then we'll end it there on the beginnings of protein synthesis. So again, messenger RNA, that's what comes out of the nucleus, and it goes to the ribosome, and the bases of DNA, they form a triplet. You know, a GU or a TC, something like that. And that's the sequence. They come in codons of 3. And that's what builds the amino acid. So this is kind of just a cartoon of what's going on. The messenger RNA is that strand, like the ticker tape going through the ribosome, small and large units. And then the proteins are what shows up after the polypeptides are made. So this is basically showing you the triplets from DNA, and then look how it pairs AUG, right, to TAC. And methionine is the start codon, so that it's a miracle how this happens. And then there's a stop codon, and it's always the same. So it knows just exactly how to pair these 3 bases to form these, and these are all amino acids. 3, 4, 5, 6, 7, 8, 9, 10. So basically, these look like the amino acids that we make in our body, not the essential ones where we need from our diet, from Popeyes, if you will. And you don't have to memorize all this. You can remember methionine as being an amino acid, but this is the magic, how it happens over and over through these triplet pairs, and you can see how they pair. TAAU, the messenger RNA is here, and here's the template. So the DNA splits through enzymatic use, and then messenger RNA is copied. And each one of these 3 triplet codons could be a template for all these amino acids. So different combinations of amino acids make different proteins. So like I said, some we can't make in our body. We need to eat to get into our system to make full proteins. That's why it's challenging when you're a vegan, but you could do it pretty simply by mixing the right foods that contain the right amino acids to get your daily doses of protein. It's not a hard thing, and it never will be. So being vegan is actually a great thing to be, as far as inflammation and obesity, diabetes. As long as it's done right, it's probably something we should do overall. But we're not going to do that because we love Popeyes right now. So we'll talk about that later. Okay. So transfer RNA, just describing that more. It bends and it brings in the amino acids, the appropriate amino acid to build. And you don't have to know the amino acid transfer synthase enzyme because there's so many enzymes you would have to learn. But know what I've been telling you, that messenger RNA brings the information to the ribosome, the copied DNA. Transfer RNA brings the amino acids. Ribosomal RNA is really what the ribosome is made of, and that's built in the nucleolus inside the nucleus of the cell. So this is really good because it shows you messenger RNA moving through the ribosome and the transfer RNA bringing in the amino acids, building each codon. Each codon triplet will represent the machinery building a amino acid. So the amino acids are attached together via these covalent bonds, which are known as peptide bonds, and then dissociated from the tRNA because they're building a protein. Like in the ticker tape thing I showed you, if you understood that. Okay, so messenger RNA loses its amino acids and goes back to do another job. And then there's a stop codon, and that's the miracle. Like, how does that know when to stop? And that's the information coming from the DNA that we found out. Okay. So building polypeptide chains. I think I told you my definition of a protein is a polypeptide chain of more than 50 amino acids. Chaperones, they help the interaction. There are other proteins that make sure that these proteins are built correctly. Kind of like a transcription factor, but it's its own type of molecule. So there's a nice close-up of how this messenger RNA goes through the ribosome. And the transfer RNA brings the triplets that make up different amino acids to build the protein. Protein synthesis. A lot of schools are closing tomorrow. You guys get a call yet? So here's a sequence. Here's the messenger RNA in there. You see the ribosome. This is in the RER, of course, where this happens a lot because this has got ribosomes, so it's protein synthesis is happening there. And this is like a primary structure. All amino acids bound together with peptide bonds. And there's disulfide bonds between some of these as well. You don't really have to know that for us. So we got to move on now after this lecture. Okay, going on more about the Golgi complex. They're further modified and they're packaged, sometimes put together with a carbohydrate, right? Remember the major histocompatibility complex, so it forms other parts of the membrane. There's other use for glycoproteins. And it depends where they're going. So it's like a post office. All these things make sense, right? The post office of the cell. Okay. So next exam, we'll start with cell division. I think we'll do, it's a very short period, cell division, and you're getting this in the lab. So I think we're going to end there for this exam and start with cell division next time because that's really quick. And again, I know you're doing that in lab.